The two or three as more as functions of the plasma membrane that is concerned as with the syllabus. One is like uh, chloride bicarbonate anion exchanger, and we have to know the carbon dioxide has that is produced during the metabolisms, particularly respiratory as processes in tissue or cell. From as cells, it diffuses to extracellular fluids, and so finally it comes into the RBC on erythrocyte that has transport of the carbon dioxide, and as the carbon dioxide diffuses due to its non-polarity in the cytoplasm of the RBC, where it combines with the water and also form H2CO3. H2CO3 कैसे होगा बेटा? H2CO3, and this H2CO3 that must be entered into the lungs, and lungs has chloride and has a bicarbonate exchanger that is known as AE, anion exchanger. And in case of human has three kinds of anion exchanger. The very A, and that is it has been designated as AE1, AE2, and AE3. AE1 it is present in the RBC, whereas AE2 that is present in the lungs, whereas AE3 exchanger that is most important, that is also present in heart, brain, kidney, etc. And also the AE anion exchanger that consists of 12 single polypeptide chain that spans the plasma membrane has 12 times. And the carbon dioxide that enters inside the lungs tissue, where it combines with water to form carbonic acid in presence of enzyme that is called as carbonic anhydrase. Or you know कि carbonic anhydrase, that is the abundant enzyme that is present in large quantity. On one point has so important is कि carbonic anhydrase has so that contain zinc in its active has a site, so that the zinc is required has so for the functions of the carbonic anhydrase. And सेकंड क्वेश्चन इसमें और भी पूछता है कि इस कार्बोनिक एनहाइड्रेज जो आपका इंजाइम टर्नओवर नंबर ऑफ इंजाइम that is higher in case of human as a body or other, that is aerobic organism, that is the carbonic anhydrase. In case of human beings, when enzymatic turnover number of the enzyme carbonic anhydrase is much higher than that of the other enzymes. And this also bicarbonate has so splits into protons and bicarbonate, and so further this also again must be as enters into the RBC, particularly in form of CO2 that must be exhaled. So this also bicarbonate and protons that further combines in presence of same enzyme and has formed the CO2 and H2O, or CO2 also goes out. Another type that is the को बताना था in last classes, as we have given you a topic that is the sodium potassium ATPase has a pump. And so the pump always the functions of the pump in a cell membrane is coupled as to the ATP hydrolysis. And the type of ATPase has so that is present in entire living organism from very beginning life as E. coli, your prokaryotes, to human as beings, including as plants, that is ATPase, and there is three kinds of ATPase. P type ATPase. बाबू पेन कहाँ है? पेन. P type ATPase that is concerned also with phosphorylations of the enzymes. F type, and F type that is also concerned with the flow of a proton. It is also most abundant in mitochondria as well as in chloroplast. Third has a V type ATPase. It is also present in vacuoles like as a lysosome, and as a vacuole, contractile vacuoles, and other as a vacuole that is just produced during the uh, breakout of the Golgi body or endoplasmic reticulum, etc., including as endosomes or lysosome anyway. The all type of the ATPase that consists of 9 polypeptide chain of 3.
डॉक्टर साहब डिस्कनेक्ट हो गए हैं क्या?
हाँ, कोई किसी तरह की वॉइस नहीं है। आई थिंक सर डिस्कनेक्ट हो गए हैं।
उनको बोलना पड़ेगा एक बार और स्क्रीन शेयर और कर लेंगे।
1 मिनट में कॉल कर रही हूँ, आप वेट कीजिए।
जब तक कनेक्ट हो रहा है मैं कनेक्ट कर रहा हूँ क्लास में, ठीक है? मेरे को होस्ट दे दो जरा, मेरे को होस्ट ट्रांसफर कर दो बोल।
वो जस्ट कनेक्ट हो रहे हैं सर।
तब तक मुझे होस्ट ट्रांसफर करना जरा।
हाँ, मैं कर रही हूँ जस्ट। सर मुझे लग रहा है कि इनका मेरे पास मैसेज भी आया है, काफी नेटवर्क इशू है।
चलो मैं
आप
कनेक्ट, मैं ले रहा हूँ क्लास, मैं ले रहा हूँ,
सेशन
मैं ले रहा
रखिएगा, हाँ इंटरेक्टिव सेशन रखिएगा। कोई
हूँ, तब तक मैं ले
टॉपिक
रहा
प्लीज आप मेजर टॉपिक स्टार्ट ना करें।
हूँ। हाँ
इन
मैं एक
लोगों
छोटा
की
सा ले रहा हूँ,
क्वेरीज
ठीक
हैं तो उनको रिजॉल्व करा दीजिएगा।
मैं ले रहा हूँ, मैं ले रहा हूँ। ओके। आप लोगों को मेरी स्क्रीन शेयर कर रहा हूँ, मैं स्क्रीन शेयर हो रही है, बताइएगा। व्हाइट बोर्ड मेरा शेयर हो गया है।
नो।
आपको मेरा व्हाइट बोर्ड शो हो रहा है, नहीं हो रहा?
नो सर।
आई थिंक सबको शो होना चाहिए, काफी लोगों, किसको शो नहीं हो रहा है? नहीं हो रहा शो किसी को भी? मेरा व्हाइट बोर्ड शो नहीं हो रहा है?
नो सर।
सर हो रहा है शो।
हो रहा है शो, ठीक, चलिए। देखिए आज हम, मैं एक छोटा सा टॉपिक ले रहा हूँ, लेकिन ये टॉपिक उसके लिए है कि जो आपके क्वेश्चंस आते हैं न्यूमेरिकल बेस्ड आते हैं, बेंडिंग पैटर्न बेस्ड आते हैं, इसको और आगे बढ़ाते हैं। अच्छा, हमने क्लास पहले ली थी, इस क्लास को हम थोड़ा सा और आगे बढ़ा रहे हैं। जैसा मैंने पिछली बार भी कहा था कि हमको दो चीजें याद रखनी पड़ेंगी अगर हमें न्यूमेरिकल क्वेश्चंस में करना है, या जो जेनेटिक्स मॉलिक्यूलर बायोलॉजी क्वेश्चन, जिन पे बड़े बड़े क्वेश्चन आप देखेंगे, चाहे वो म्यूटेशन से रिलेटेड है, चाहे वो डीएनए से रिलेटेड है, आपको फ्रेगमेंट को देख के उनकी बेंडिंग पैटर्न एनालिसिस करना, जेल इलेक्ट्रोन मूवमेंट करना, उनका आइसोइलेक्ट्रिक पॉइंट करना, कौन सी प्रोटीन किधर से किधर मूव करेगी, ये सारे सवाल होते हैं, है ना। मैं आपको लास्ट क्लास में मॉलिक्यूलर स्ट्रक्चर ऑफ नाइट्रोजन बेसिस डिस्कस किया था क्या? नाइट्रोजन बेस का मॉलिक्यूलर स्ट्रक्चर बात की थी मैंने क्या? एनीबॉडी? किसी को पता है? कोई तो बोलिए। मैंने आपको लास्ट क्लास में बात की थी, नहीं की थी?
ओके।
हेलो?
हाँ, आई एम कनेक्टिंग।
कनेक्ट।
कैन आई कंटिन्यू?
हाँ, यू कैन कंटिन्यू सर।
अच्छा ठीक है। So that sir, so I was discussing about the sodium potassium ATPase as a pump, and all kinds of the in human as of beings, all these kinds of the cells, nearly about as a muscle cells, heart cells, renal as a cells, myocytes, adipocytes that have sodium potassium ATPase as a pump, and there is three kinds of ATPase as a enzyme is as a present in as a living cells. The very first kind, that is the F type ATPase, P type ATPase, and V type ATPase. F type ATPase, so that is concerned with the flow of as a protons, and so this kind of as a F type pump that is present in most abundant form number in as a chloroplast and in mitochondria. That is as a during metabolic processes, yeah, electron transport system, proton is flow. Then as it is captured by ATPase as a pump for the phosphorylations of the ADP with inorganic phosphate to form ATP. The P type ATPase so that is coupled also with the export of, yeah, outflow of the sodium ions and inflow of the potassium ions. And the ATPase as a pump that is present in the myocytes, epithelial cells, renal cells, and cardiac cells, etc. It has to consist of the 9 polypeptide chain of as of 5 types, namely 3 as alpha polypeptide chain, 3 beta polypeptide chain, and 1 molecule each, delta, gamma, and epsilon. The delta, gamma, and epsilon that remains embedded in the lipid bilayer, whereas the alpha polypeptide chain and beta polypeptide chain that has a passage toward the cytosols. As has an influx of the sodium ion that raise its concentration inside the cytoplasm, and as a potassium is compensated by its outflow through the plasma membrane. But however, the sodium ion must be remain outside the cell, and potassium ion must be remain present inside the cytoplasm. So that as concentration of sodium ion increases in the cytosols, then the sodium potassium ATPase pump that becomes active. As concentration of sodium ions increases in cytosol, then also the enzyme sodium potassium ATPase that faces toward the cytoplasm that has 3 sodium ion as a binding sites, whereas it has allowed the binding of 2 potassium ion due to there is 2 regions. One region, the atomic number of sodium ion is 11 and atomic number of the potassium ion is 19. And so the size or atomic size of the sodium ion is generally about 0.98 angstrom, whereas size of the potassium ion that is nearly about 1.32 something angstrom. As the sodium approaches the enzymes, the sodium enzymes undergoes conformational change, and so the ATP binds with the enzymes, and so the aspartate amino acids that remain present in the active site of enzyme that undergoes phosphorylation with the hydrolysis of the ATP, and as ATP change into ADP and inorganic phosphate. This inorganic phosphate attaches to the carboxylate anions of the sodium aspartate residue and phosphorylated sodium enzymes that allows the binding of sodium ions, but however it has reduced affinity with the sodium ion. As enzymes get phosphorylated along with sodium ions, then it inverted toward the extracytoplasm, and the affinity of the sodium potassium ATPase pump in dephosphorylated form has affinity for the sodium ion. Whenever it is phosphorylated as sodium ion binds with the enzyme, then it has reduced affinity with the sodium and it has much affinity with the potassium ion. So that as a result, 3 sodium ions moves along with the sodium potassium ATPase pump toward the extracytosolic sites, and so it loses 3 sodium ions, and later one phosphorylated enzyme has much affinity for the potassium ions, and so that 2 potassium ion binds with the enzymes. As potassium get bonded with the enzymes, then it get inverted toward the cytosolic sites, and so in case of the cytosolic site, there is another enzyme present known as phosphatase. The phosphatase also catalyzes the dephosphorylations, so that aspartate residue that was phosphorylated in earlier that get dephosphorylated, and dephosphorylated enzyme has less affinity or has not affinity for the potassium ion. That's why enzyme loses 2 potassium ions. So that this process which takes place all the times to must be cell brings to at equilibrium, and sodium ion that goes outside and potassium ion comes inside. And one question, why 3 sodium ions goes out and 2 potassium ion come in? There is 2 reasons. One reason, the size of the enzymes that allow the sitting of the 3 sodium ions, and as it allows the sitting of the 2 potassium ion due to their atomic size. But most important, the cell has to maintain the electrochemical gradient. To maintain the electrochemical gradient, the positively charged or cations must be exported outside to maintain the negatively or polarized conditions inside the cells. And as potassium comes, 2 molecules of potassium comes, and as of nearly about 40 to 50% of potassium ion that bound with the organic molecule, organic proteins. So that has potassium ion, even though potassium concentration of potassium ion inside the cell is higher than that of the other ions, but in spite it does not generate the positive charge, because the majority of the potassium ion remain bonded with others. So the sodium potassium ATPase. Second, in case of heart, myogenic has a functions. Particularly, one has a drug has been isolated from has a plants Digitalis purpurea, and so the drug is named as digitonin. It is cardiotonic steroids. In case of the cardiotonic steroids, yeah, people who is suffering from uh, reduced as a contractions of the as a heart, then as a cardiotonic steroid is given, and this cardiotonic steroid is given, then it causes the depolarizations of the cardiac cells due to the influx of the sodium ion, and sodium ion has an influx. As sodium ion goes out, in spite of the sodium ion, there is influx of the sodium ion goes out, and calcium ion comes in. Then calcium ion is required for the muscle contractions so that the cardiotonic steroid is also, and that regulate the ionic movement and maintain the ionic gradient of the cardiac cells. Second, another kind the most important the transporter in that is known as ABC transporter. And ABC has a transporter, or another example that is the chloride channel. The first of all, ABC transporter. The full form of ABC is the ATP binding cassette. This ATP has a binding cassette that consists of the 12 membrane helices has a proteins. And so the ABC transporter that consists of 12 helices membrane with 2 molecule of NVD. NVD stands for nucleotide binding domain. So that the ABP or another it's similar has a pump, similar has a proteins that is known as aquaporin. Third, it's similar as a proteins that is the chloride has a channel. So there is 3 examples that is related with this 12 membrane helices protein. Another 4th example that is the bacteriorhodopsin has a protein. Bacteriorhodopsin has that is present in aerobic bacteria, and this aerobic bacteria that utilizes the flow of protons for generating the ATP rather than photosynthesis. This rhodopsin has proteins that is called as bacteriorhodopsin protein, not rhodopsin that is present in eye, but however it is the bacteriorhodopsins that is present in salty marine bacteria, where there is a flow of a proton that allow the bacteria to generate the ATP. So that 3 or 4 examples that is related with 2 12 membrane helices, because this 12 membrane helices has proteins that consist of 6 helices and another 6 inside in 6 helices that has 1 molecule of NVD, another 6 helices that also has 1 molecule of NVD. In a rest condition, it remains in closed conformations. But whenever, for particularly example, I am giving that is a case that is a cystic fibrosis. I am giving one examples about the cystic fibrosis. Cystic fibrosis that is more prevalent as to disease in America or other countries, because in as biotechnology was progressed. That then in 1989. Then the genome protein was decoded about the chloride channels. Then the writer has written there is a glimpse of the sources for a curing of the chloride shift, because there was heavy mortality, death in USA in comparison to other kind of diseases, so that much heavy mortality due to the failure or treatment of the chloride channel in comparison to cancer and other diseases. So that the chloride channel mostly present in the epithelial tissue, lungs, and testes, etc. And most prevalent that is in lungs, because lungs has to export the bicarbonate chloride channel, just as before I was sharing. So the lungs has bicarbonate and chloride, the chloride has channels that as a concentration of chloride ions that increases, then it has negative charge. That's why it attracts more has a positively charged like as a sodium ions. So that has a defective chloride has a channels that has a, does not allow the export of the sodium ions. As a result, has a, the lung cells it has higher concentration of the sodium ion, and as a concentration of sodium ion increases inside the cell or ionic concentration increases, then has the lungs tissue or the air passage of your windpipe of the respiratory has to tract that is lined with the mucus, so that mucus contain the of water, and also so that has the water has from area tracheal has a passage that influx inside due to the osmosis, and so trachea has to get dry up. And whenever any has a allergic response has a takes place during has a coughing has a starts, and as a due to heavy as a coughing, but as a cough does not expelled out. That's why sometimes the aerial respiratory has a tract that has to get damaged, and this has to damages as a respiratory tract that provides the fertile area of the harmful as a bacteria like a pseudomonas coccus and pseudomonas aureus. These are two bacteria has to damage the respiratory has to tract, and finally the others. That's why the chloride channel has to it is mostly, if it was genomic amino acid sequences was observed, that then also at the 506 and 507 and 508 position there is repeating same amino acid, that is the phenylalanine, or there is it has been reported that any has a absence of the phenylalanine either at 506 position or 507 position or 508 position, that then it has causes the cystic fibrosis. So one example is here. Second example, that is the I am giving aquaporine. Aquaporine, there is just more than 10 or 10 kinds or 12 kinds of aquaporine is just present in the humans. But one kind has aquaporine has to we are taking example aquaporine 2, that is as a present in renal tissue, and as we know renal as a tissue like as a collecting duct that is also known for the reabsorptions of the water, and as a reabsorption of water is facilitated as a by the or influenced as a by the another hormones that is known as antidiuretic hormone that is released from पोस्टीरियर पिट्यूटरी. हम जानते हैं पोस्टीरियर पिट्यूटरी से निकलता है. और जैसे ही देयर इज क्वेश्चन कि डायबिटीज मेलिटस एंड डायबिटीज इंसिपिडस, तो सो दैट इज डायबिटीज इन प्रीवियस बुक्स और दैट सेज दैट कि डायबिटीज इंसिपिडस इज काउज्ड ड्यू टू द deficiency of the hormones known as antidiuretic hormone. But as genetic information, applications of the biotechnology and others that says the diabetes as insipidus, that is that due to the defect in the receptor that has received the antidiuretic hormone or ADH hormone. So that is a collecting duct. This as a collecting duct as so that contain the as a receptor as a for the antidiuretic hormone or ADH, and this as a ADH hormone as a comes, but as a receptor is defective because it is similar to the GLU24 in case of the muscles I have discussed in last classes. So that's why the as a defective as a receptor does not allow the formation of second messenger like cyclic AMP, and as this so that as the failure or concentration as a concentration of cyclic AMP is lower, then the barter has a channels. Aquaporine that is concerned with the channels of passage has to provide the movement of the barter, so that as the channels aquaporine has does not get open. That's why barter is not has reabsorbed. That's why so that as there is a lack of the has a barter in as a body, and in spite has this that has a barter that has a goes out along with the urine, that is called a diluted urine. If a antidiuretic hormone is there, but however there is defect in the receptor or has a channel like aquaporin, then it causes diabetes insipidus. Second example. Third example, in case of the bacteriorhodopsins. This is the bacteriorhodopsin, that is a 12 membrane helices protein, and it is mostly first it is as a present in the Dead Sea. Dead Sea we know कि वहां पर salt का concentration इतना ज्यादा है कि ऐसे because no one other organism can survive due to the higher salt concentration. But also the few bacteria is able to also survive because so whenever it comes to the above water surface, then it traps the radiant energy, or that this radiant energy that has to convert the bacteriorhodopsin. Bacteria is nothing but I am telling rhodopsin that is present in our eye also, human eye and other other animals eye also. Then they has bacteriorhodopsins that has splits into the two kinds of component. One is protein, that is the opsin. Second one is the retinal. We know retinal is the prosthetic has a group of the rhodopsins, or we know it is the aldehyde form of the functional vitamin A. And so this as a retinal undergoes 13 cis conformational changes. In case of animal has eyes, it undergoes cis to trans conformational change, but in case of the bacteria, it also undergoes the 13 cis trans conformational change. At the rest, it is present in the cis configuration, the cis conformation, but under the influence of the light, it undergoes the 13 trans conformational changes. As the light, it received as a light, then the conformational changes takes place in the retinal or opsin has a protein, then has a opsin that allow the movement of the proton, a flow of the proton, and this flow of proton is finally captured by the ATPase enzyme, and this ATPase phosphorylate the ADP with inorganic phosphate and form the ATP, so that this bacteria, in spite of receiving the glucose and other molecules to generate the ATP for their vital functions or physiological functions, they also generate ATP in presence of the light. Yeah, the many examples. And last one is me, in case of the plasma membrane, now has to be moved toward the receptors. हालांकि एक प्रॉपर टॉपिक है सेल सिग्नलिंग्स में. Receptors, that is the integral membrane has a protein, transmembrane has a proteins, are nearly what the 2% of the total plasma proteins that is represented by the receptor has a proteins. So many kinds of receptor has a present in cells, but however the well-studied has receptors. Well-studied as a receptor like the adenylate cyclase, guanylate cyclase, but the effector as a molecule is different in different as of cases. If example as a like glucagon hormone, we know that glucagon hormone, it is also known as hunger hormone. Yeah, you can say hunger response. Whenever as a blood glucose depletes beyond the normal value, then as the alpha cells of pancreas releases glucagon hormone. Or if we has to intake the tea or caffeine, then has tea or caffeine also stimulate the alpha cells of pancreas to release glucagon hormone. The glucagon hormone is much responsive to the hepatocytes, and the hepatocytes, so that the glucagon hormone that travels along with the blood flow, finally it reaches to the hepatocytes, and the hepatocytes has its receptor known as serpentine receptor. Serpentine means it has spans the membrane 7 times. It has crossed the lipid bilayer 7 times, and its exterior parts that has an amino terminus while its cytosolic site has carboxylic terminus. And so the extracytosolic side as it received, because as the glucagon hormone that consists of 29 amino acids, and any as a protein does not as a crosses the plasma membrane, so that as a protein that mediate its physiological functions and influence the cell physiology through receptor present on the plasma membrane. So that as hormones binds with the receptor, that then it make the hormone receptor complex. As hormone receptor complex is also formed, that then as the receptor is coupled also with the specialized as a protein known as G protein. So that the cytosolic side of the plasma membrane has G as a protein, that is again integral membrane has a protein. As it get activated, or if one point is here, G protein, what is G protein? The G stands for guanosine nucleotide binding domain. It either has to contain GDP, it can also allow the binding of either GDP or GTP. Whenever cell is in rest, then it has GDP in its active sites. But whenever the hormones binds with the receptor, then the G protein get activated, or G has a protein that allows the binding of GTP. GTP replaces the GDP, but however cytosolic side contains one more enzyme, one proteins that is known as G protein, G exchanger protein. G exchanger protein, this as a protein and allow the carry the GTP, and it attaches the G has a protein particularly to the alpha subunit, and alpha subunit also has GDP in active sites, but as a GTP is replaced by the GTP, as a GTP binds, then the G protein, that is the trimeric has a protein. This has a trimeric protein consists of one molecule of alpha, one molecule of beta, one molecule of gamma, in which also the molecular weight of the alpha subunit that has 42 kd, whereas the beta subunit that has 38 kd, and so gamma is a protein that has 7 kd Dalton. So that alpha only alpha has physiological processes, hormonal response. In rest condition, it has GDP. As a hormone binds to receptor, then it get activated. So that has a G exchanger as a protein replaces the GDP by GTP. As a GTP binds with the alpha, then also it dissociates from the trimeric subunit, and also the beta or gamma remain there, and also alpha moves from that, and also this alpha carry with the GTP toward the effector as a molecules, and this effector molecule is known as adenylate cyclase. The adenylate has a cyclase that as it get activated, then it catalyzes the cyclization of the ATP into cyclic AMP. Cyclic AMP, so that ATP is hydrolyzed into AMP plus PPI, and so the PPI is further hydrolyzed by inorganic phosphatase enzyme into 2 molecules of inorganic phosphate, and involving nearly about 8.6 kilocalorie energy. This 8.6 kilocalorie energy that catalyzes the cyclizations of the AMP into cyclic AMP. So that this cyclic AMP is now known as 3' and 5' adenonucleoside 5' monophosphates. This as a cyclic AMP serve as a second messenger, so that hormone has that is first messenger, and cyclic AMP that is generated in cytosolic sites act as a second messenger. This as a cyclic AMP now activate the inactive enzyme that is already as a present in the cytosol, that is known as protein kinase A. This as a protein kinase A that has becomes activated or get phosphorylated, that the active as a protein kinase A that activate the enzyme that is known as phosphorylase enzyme. So that as a phosphorylase enzyme becomes active, then as a phosphorylase catalyzes the phosphorylasis of the glycogen into glucose 1 phosphate, further another enzyme, that is the phosphoglucomutase, that catalyzes the isomerization of glucose 1-phosphate into glucose 6-phosphate. Later one, the phosphatase enzyme so that remove the phosphate from the glucose 6 carbon and finally generate the glucose. So that has the glucose as a molecule that is also produced by the glycogenolysis in hepatocytes that diffuses as through the in the blood vessels, and so that has a blood glucose concentration become higher and higher, and as it supply then has this glucose to the brain and other as a muscle tissue and others, so that माने the ग्लूकागोन या कैफीन, जैसे हम सुबह कहते हैं चाय पीने से फ्रेश महसूस करते हैं, तो वहां पर जो चाय और कैफीन है ना, दैट एक्चुअली हैज सो कॉजेज द रिलीज ऑफ द ग्लूकागोन, एंड ग्लूकागोन हैज सो हार्मोन दैट causes the glycogenolysis through this as a process. Even though the people who are not receiving the as caffeine or chai, tea, then the in stress conditions, yeah, in hunger condition, hunger means glucose, blood glucose consumption declines. That is the hunger situation, because it has been correlated as with the bacteria कि एक बैक्टीरिया को अगर ग्लूकोज और लैक्टोज दोनों देते हैं तो ग्लूकोज जैसे कम होता है तो बैक्टीरिया कम्स इनटू द stress, that is known as hunger. That then has so lactose metabolism start through the lycopron has a genome and lycopron systems. Another topic. So that so this so you, if you see has the evolutionary has a response from us, so the bacteria to human as so kinds the whenever glucose, because plasma membrane or E. coli has so that is so E. coli that prefers more has so glucose in region in comparison to other as so hexose like as a galactose and others. So that the so in case of human, yeah, so higher has so vertebrates or vertebrate, the plasma membrane only allow the influx of the monosaccharides rather than disaccharides, so that the cell prefers glucose for its further catabolic processes. That's why the glucagon that releases the glucose has so molecules through the glycogenolysis. Same has so actions that is carried out by the epinephrine has so molecules. The epinephrine that is released also during the stress condition, but however the epinephrine function, their function is same, but has so it act upon the muscles myocyte. एक क्वेश्चन जनरली आता है कि अगर ऐसा किसी व्यक्ति को अगर हम कैफीन या चाय पिलाते रहेंगे तो क्या होगा, तो वहां पर एक क्वेश्चन कि और कैफीन पीने से या चाय पीने से भूख क्यों नहीं लगती है. So there is two has a continuous question. The two questions. If as a caffeine is continuously as a given, then has the cell has a membrane, yes, responsive as a cell, a hepatocyte cell get has the sensitized. So that if as once time 10 minutes later, 15 minutes later, repeatedly as a caffeine is given, then new repeat of the as glycogenolysis, a new process of glycogenolysis start. No. So that as if already as hepatocyte have received as the message, then so the glycogenolysis continuously take place. Secondly, as epinephrine as a second question, so the caffeine that is so contain the inhibitor as a molecule known as phosphodiesterase. So that as a concentration of cyclic AMP increases, so that has so this phosphoactivity of the phosphodiesterase is inhibited due to the caffeine molecule. As a result, cyclic AMP concentration remains higher in cytoplasm, so that glycogenolysis continuously takes place. That's why so the blood glucose level remains higher. If also the caffeine is not also there, the phosphodiesterase has so enzyme has so that normally hydrolyzes the 5' 3' cyclic AMP into 5' AMP. So the response is over, glycogenolysis is now over. So that has so the another topics. If you looking, sir. आवाज सुनाई पड़ रही है? हेलो,
यस,
हेलो मैडम, हेलो, आवाज आ रही मैडम?
यस सर.
है ना। तो एक मैंने जनरली प्लाज्मा मेम्ब्रेन का और सिलेबस में अगर कुछ और है तो मैडम कंक्लूड कर देंगे, अदरवाइज हम दूसरा टॉपिक शुरू करेंगे, माइटोकॉन्ड्रिया। सुन, कैसी जगह बेटा तुम्हारा? सब कुछ तो है ना। हाँ जी मैडम, हेलो, आवाज आ रही मैडम? अनम्यूट करना, क्या करना है?
सब कुछ ठीक है,
ये अनम्यूट करें,
सब कुछ ठीक है, कुछ ठीक है। अनम्यूट आप।
इस चार्ट में लिखें।
Okay, sir.
ओके सर, नेक्स्ट क्लास में मैडम, है ना, माइटोकॉन्ड्रिया आई विल ऐसा कंटिन्यू टुमारो, और जब व्हेनेवर यू विल प्रोवाइड मी, गिविंग मी अपॉर्चुनिटी। थैंक यू मैम। कॉल कैसे करो, कॉल कर लूं
किसको?
जयरा मैडम को, यहाँ से आएगा ना, ऐसे यहाँ से।
मीटिंग को लीव करना होगा। मीटिंग
मीटिंग का नहीं होगा,
ना,
यहाँ से नहीं होगा इसे करके।